Hello AP Bio Bruins, and this is Mrs. Farouk, and we'll talk about electron transport chain. Now, in order to understand the electron transport chain or oxidation phosphorylation, one must remember what is facilitated diffusion. That's passive transport using a protein channel or protein gate. The energy coupling concept, the idea that when you have negative G or free energy, we couple that to make something that does require energy. And the redox, what's a redox reactions? And remember the mitochondrial structure and that demo we did in class, that the mitochondria is like two bags. You have the outer membrane, and then in between the second bag and the first bag you have your intermembrane space, and then inside the second bag is where you have your matrix. So just quickly reviewing the overview of cell respiration, we start off with glycolysis outside of the mitochondria. And remember, glycolysis is taking that 6-carbon sugar and splitting it into a 3-carbon molecule pyruvate. And then that pyruvate, when you have oxygen, will then go into the mitochondria. And this only produces just a little ATP, 2 ATPs, and we produce a electron carrier that carries the energy NADH, which will then go to the electron transport chain, ETC for short. So that 3-carbon pyruvate goes to the intermediate step, and that 3-carbon pyruvate becomes a 2-carbon molecule, releasing carbon dioxide, making a little more of NADH. And then that intermediate step, the acetyl-CoA, enters the Kreb cycle. And then in that Kreb cycle, glucose is completely oxidized, releasing the byproducts as carbon dioxide, making a little ATP. Remember, this goes through 2 cycles per glucose molecule, so it makes 2 ATPs, and a lot of NADH, and then a second player, a second electron carrier called FADH2. And all this electron carriers that picked up all this energy will now go to the electron transport chain and make a lot of ATPs. And the oxygen, this is where oxygen is needed, is the last electron acceptor, and it will be reduced and become water. Okay, so once you have all that NADH and FADH produced in those other processes, it comes to the electron transport chain. Now, the electron transport chain is composed of series of proteins, which are integral proteins and peripheral proteins. You also have a lipid, where the electrons are passed from one protein to another. So there's a series of reduction and oxidation reactions. All right, and when something is reduced, it's picking up that energy. So that energy is coupled. So here you have NADH, which is oxidized, and here you have a protein that's reduced. So when it picks up that electron, it's picking up energy. That energy is coupled to move the hydrogen ions from the matrix of the mitochondria into the intermembrane space. So as these electrons pass down the electron transport chain, hydrogen ions or protons are being transported. Okay, and then what's happening is you're forming a high hydrogen gradient in the intermembrane space. So you have a high concentration of hydrogen ions in the intermembrane space, which will further, from this, what we're doing is we're establishing this gradient or proton electro gradient and high proton gradient, which will diffuse to facilitate a diffusion through this protein, integral protein called ATP synthase. So it's an enzyme, and through hydrogen it will provide that energy to catalyze or hydrolyze ADP to ATP, and that's what's called oxidative phosphorylation. Okay, so basically what's happening is we establish this high concentration of hydrogen in the intermembrane space used by the energy provided by NADH, by reducing NADH and FADH. And basically this is what we call chemo osmosis. So the hydrogen, through facilitate diffusion osmosis through this enzyme, and by its energy, it's coupled, this enzyme goes through a series of conformational changes and catalyzes ADP plus phosphate to ATP. Okay, so it sort of acts like a motor here. So if you want to get further details, look at this animation. So this is another way of looking at here. The green space is your intermembrane space, and here this brown is your matrix of the mitochondria. So your electron transport chain is basically a series of proteins on the intermembrane space. So again, when NADH is reduced, when it's oxidized, it reduces the electron transport chain protein. It picks up that electron, and as that's reduced, that energy is used to transport hydrogen into the intermembrane space, and thus establishing a high concentration in the intermembrane space. So as this hydrogen protons are pumped into the intermembrane space, we establish a high concentration of gradient in the intermembrane space. Okay, once that happens, now that hydrogen has formed this electron, this proton electrical motive force, and this is where we can start having chemo osmosis through the ATP synthase. And then the very last electron acceptor is our oxygen. Okay, so without oxygen, you don't have that pull, that electrons passing from one protein to another. And when oxygen accepts that last electron, it becomes water. Okay, so then finally, when you do have that hydrogen concentration, hydrogen ions diffuse or osmose through this ATP synthase and hydrolyzes ADP plus inorganic phosphate to ATP. Now ATP synthase is really neat. We also see this in bacteria, and we also will see this in chloroplasts. Okay, so here's a summary of oxidative phosphorylation. NADH and FADH are oxidized, and those electrons is passed from one protein to another, and as it's being passed on, pumps the hydrogen ions into the into the intermembrane space, creating a high hydrogen concentration gradient in the intermembrane space, which is then used to provide energy. And here you see the energy coupling, where when hydrogen passes through the enzyme, it has a lot of negative delta G, so that energy is used to hydrolyze ADP plus P to ATP. Okay, there's just a little animation just to help you kind of visualize this. Okay, so again, the hydrogen gradients going through the ATP synthase, and that makes the ATP go through conformational changes that allows it to catalyze ADP plus P to make ATP. And notice this energy is being released slowly in small steps, and the cell harnesses these small amounts of energy to produce ATP. And again, down here you see the oxygen as the very last electron acceptor. So as the electrons are being passed from one protein to another in the electron transport chain, you can see the energy falling down. So use this as a graphic organizer to review electron transport chain, right? What are the reactants? What are the products? Where is it happening? And what's the purpose of the electron transport chain or oxidative phosphorylation? And remember what's happening here. We're harnessing that free energy into small steps. So life is like this picture, where the delta G is coupled pumping that hydrogen ions into the intermembrane space and harnessing that energy in small multi-steps, not just one big step like here.